Today we'll be doing microbial extraction from aphids of apple. So first you have to focus on it. So first you have to use PBS. Tissue paper. So whatever you bring inside the food should be sterilized first. So we'll take 70% ethanol for surface sterilization. Mix it. Okay, so we want to sterilize the surface. Balance है balance. एक और अपना डाल दो. Okay, so we'll spin it for a while. Just give it a short spin. So this will settle your aphids at the bottom. So we'll have to use it again. So your aphids are now at the bottom. They are settled, and we'll throw the ethanol. Okay. So what you do with the same tip, you can slowly discard the so you have to be careful you don't disturb the aphid pellet, otherwise you'll lose. Okay, this much is enough. So we have discarded ethanol. Now we will sterile distilled water. So we will add sterile deionized water or distilled water, whatever is available. And again spin it after shaking. So this is a short spin because they are very heavy, so you have to only give a short spin. So now Okay, so you don't have to lose your aphids. So now I will give one more wash. Okay. So you have to ensure if there are any contaminations in your water, so you have to autoclave everything. You have to clear your water, autoclave your tips, autoclave your appendix, because we do not want that any bacteria from outside should come. Okay. So now we have this, so we'll sterilize our. Okay. So this is your central ethanol. We'll sterilize this is a small vessel, a mini grinder. Okay. So I'll try to burn the ethanol, but you don't have to burn it. Okay. So now I'll open it and slowly try to grind it. Aphids. Okay, and we have what we have done. We have sterilized its surface so that all the bacteria that are outside will die, and give it a wash, and then they'll be washed off. Now the aphids inside won't die. The microbes inside will not die. So once we slowly puncture these and grind these. Okay, now we'll pour this solution on your petri plates. Overall petri plates. Okay.
Petri plates.
Close up. So how much we have to grind? So these all should rupture. These aphids, you can see they're all fragmented, and all the gut is outside, and now we want to see what's inside. बाहर नहीं खोलना, हमेशा जब ऑटोक्लेव्ड हो सबसे अंदर होता है काम, बाहर नहीं होता है, बाहर होता है कंटामिनेशन आ गई. ठीक है. So this is not good autoclaving. You can see you have vapors. That means when you have washed it done with autoclaving, wet autoclaving, so we can put them in a dry heat oven at 300 degrees centigrade for 4 hours. Wet heat autoclaving we do for almost 20-30 minutes. So when you are a pro, if you are a pro, you can use the gloves also, and you will not contaminate the flasks. So we don't need this. So 4 plates. (100 ml is enough for 4 plates. So 25 ml each. So we don't require this. So that's why I put the cotton plug down. Otherwise, if you are reusing the media, you will not put the cotton plug down. So we'll wait till this solidifies, and till then let's take a break. So when we add media, what do? We stack these plates so that the bubbles are not. See the lower plate, you don't have any bubbles, because this media is hot. It will condense the bubbles, and so you'll have a pure clear plate. But as you go up, you'll condense water, so it will not harm us. But always try to avoid this. So we'll need ethanol. ये इसको हम सीरियली डायल्यूट भी कर सकते हैं. So we can serially dilute it also. Okay. So this is ethanol. Okay. So this will catch fire, all the ethanol will burn. Okay, so that will sterilize or loop, glass loop. So we'll first cool it, okay, on the edges, and then we will spread it. So once it's thoroughly cooled, so we'll take a marker and we'll write. So we'll incubate this plate overnight, and tomorrow we'll see what bacterial cultures have grown. Okay. So this we'll store, will not throw, and then we may serially, if we find the bacterial colonies are very high, then we can serially dilute it tomorrow.